All right, so this is the last lesson that we're looking at for Unit 2 with metabolic processes. And in this lesson here, we're going to be looking at adaptations of photosynthesis. So we've been talking about how plants itself are able to create glucose, that they are self-producing organisms and they can create their own food. However, not all plants live in the same type of environment. Some live in really cold, really hot areas of the world, and because of this, it actually changes the way how they create glucose. So it kind of alters the way that they go through photosynthesis. And this has happened over thousands and thousands of years. This is not something that's just happened overnight to allow for optimum production of their glucose. So we talked about in the last unit in 4.1 and 4.2 when talking about light-dependent and light-independent reaction, especially in the independent reaction of the Calvin cycle, we talked about how carbon was fixated. So how we took a carbon dioxide molecule and turned it into something that is usable, and we turned it into glucose. So again, in this lesson, we're going to be looking at different ways that plants are able to fix their carbon fixation to allow for it to be more optimal to them. So by the end of this lesson, you will be able to explain how plants adapt to hot and cold environments and the C4 and CAM process. Autotrophs. So plants are autotrophs, which are self-producing. They're able to create their own glucose. Where does this happen? It happens in the leaves of the cell, more specifically in the mesophyll cells, and also the stoma plays a huge role in this because stoma is what allows for carbon dioxide to enter and for the oxygen to leave. The rubisco is a critical enzyme in the light-independent reaction, so in the Calvin cycle. However, it has an undesirable property. Rubisco can use oxygen as a substrate as well as carbon dioxide. In fact, oxygen and carbon dioxide compete with each other for the same active site on the rubisco enzyme. When oxygen reacts with ribulose 1,5-bisphosphate in the process called photorespiration, the products are two carbon compounds called phosphoglycate and one of the three carbon compounds, 3-phosphoglycerate. As a result of photorespiration, all of the energy used to regenerate the ribulose 1,5-bisphosphate is wasted, thus reducing the efficiency of photosynthesis. Under normal conditions, when the temperature is near 25 degrees, C3 plants lose 20% of its energy used to fix one carbon molecule. Biologists estimate that the maximum possible efficiency of photosynthesis in C3 plants, assuming each photosystem absorbs a max amount of light, is 30%. Again, we're going to be going through that whole process of what's going on in this lecture slide itself. So again, here, this is just a little drawing of what a mesophyll cell looks like. How we have our cell wall, and inside the mesophyll cell we have all of the chloroplast, that's these red dots here, that can be found. It's also important to note that we have a central vacuole and our nucleus that's present. In the Calvin cycle, rubisco forms an unstable 6 compound that breaks down into 3 carbon compounds. Since the first stable compound has 3 carbons, this is why we call C3 photosynthesis. So this is in the normal cycle process of the Calvin cycle that we've been talking about. So carbon fixation happens in the light-independent reaction. It happens in the Calvin cycle. C3 plants is about 80% of plants on Earth. So why do we call it C3 plants? Well, we call it C3 plants because normally what happens in the Calvin cycle process is that we end up creating 2 3C compounds through that cycle process itself. So that's why we call it C3 photosynthesis. So that whole time in the last lecture we were talking about the Calvin cycle, we were talking about the cycle process of C3 plants, or the plants going through a C3 photosynthesis. So this occurs in the stroma of the cells. It uses ATP and NADPH from light reactions. So from the non-cyclic photophosphorylation, it uses carbon dioxide, and it does this to produce glucose. It takes 6 turns and uses a total of 18 ATPs and 12 NADPH. 6 turns meaning it needs a total of 6 carbon dioxides 18 ATP, 12 NADPH to make one glucose molecule. So this is kind of what this image is showing you here, is the Calvin cycle process. So we have our RUBP that is going to be bonded with our carbon molecule here, thanks to the Robisco enzyme. So the Robisco enzyme is going to cleave off this carbon dioxide and attach it to our RUBP to make a 6-carbon compound, which becomes unstable, and it's going to break it apart into two 3-carbon molecules, which we also call PGA. Sorry that this isn't the nicest, PGA. Okay, they both require to break down one of these 3-carbon molecules, a total of 1 ATP and 1 NADPH. So that's why we have a total of 6 and 6 here, is because there are 6 of these 3-carbon molecules. To break it up, and then it goes through the whole process. We end up creating a glucose out of it from 2 G3Ps, and then the process goes back to helping to create the RUBP, which we need at the beginning of the start of the Calvin cycle. So this is nothing new. This is what you've learned through the cycle of the Calvin cycle, which is also known as C3 photosynthesis. So again, remember, C3 equals the Calvin cycle. Robiscos can bind oxygen as its substrate. So basically, instead of this process here where carbon dioxide carbon is being cleaved and added to this molecule here, what Robiscus can do is actually take a an oxygen molecule instead to do this. So it can also bind oxygen as its substrate. This results in the formation of a 2-carbon compound and a 3-carbon compound that are not produced, that are now produced for photorespiration. So it reduces the product that's needed for photosynthesis, and it reverses carbon fixation, because it basically didn't allow for us to go through a whole process of making these 3-carbon molecules. It kind of backtracked us. Because of this, plants lose approximately 20% to fix 1 carbon molecule. It loses 20% of energy to fix 1 carbon molecule. So this isn't really ideal, nor is it sustainable. So when does photorespiration take place? It usually takes place on hot, dry, bright days. So the stomata is close. And basically because of this, it doesn't allow for carbon dioxide to enter into the plant. Instead, it's going to use the oxygen that's found in the plant to allow for that fixation process. Again, it produces 2 carbon molecules instead of 3 carbon sugar molecules. And by the end of this process, it makes no sugar molecules or no ATP. So plants in dry environments have a hard time with this method of carbon fixation, that normal C3 fixation, because in dry environments the stomata is closed to reduce water loss. This also blocks oxygen and causes it to build up in plants. Because oxygen builds up, the Robisco will add oxygen to RUBP instead of carbon dioxide, which will then create a 2-carbon product and a 3-carbon product. So C3 photosynthesis is mostly used most of the time, but sometimes in arid environments, in hot and dry climates, it's not the most efficient. Because of this, these plants have adopted a way of life in hot, dry environments that have actually allowed them to kind of fix this whole process to make their method of carbon fixation a lot better, that helps to save water without shutting down the photosynthesis process. So again here, because of photorespiration, plants have a special adaptation to limit the effect of photorespiration. So we have C4 plants and CAM plants. So C4 plants separates the initial uptake of carbon dioxide from the Calvin cycle into different types of cells. Carbon fixation occurs in mesophyll cells, where a Calvin cycle occurs in a bundle sheath cells. Bundle sheath cells are impermeable to carbon dioxide, concentrating it in mesophyll cells, making C4 plants more efficient. It's called C4 because the stable compound is a 4-carbon malate. So, where do C4 plants exist? In hot, moist environments. 15% of plants on the planet go through the C4 process of photosynthesis. This includes some types of grass, corn, and sugarcane. It divides photosynthesis into two parts of this cell. So one part happens in the mesophyll cell, and the other one happens along in the bundle itself, the bundle sheath cell. And then, again, like I said, the light reaction happens in the mesophyll cells, whereas the Calvin cycle itself happens in the bundle sheath cells. So C4 plants have a structure that separates the initial uptake of carbon dioxide from the Calvin cycle into two different types of cells. In the outer layer of the mesophyll cell, carbon dioxide is fixed by addition to a 3-carbon compound called phosphoenol pyruvate, PEP. The product is a 4-carbon compound known oxaloate, giving these plants the name C4. The oxaloacetate is converted to a 4-carbon compound malate and transported into the bundle sheath cell. There, the malate is decarboxylized. This results in a 3-carbon compound pyruvate, which is then transported back into the mesophyll cell and converted into PEP. The bundle sheath cells are impermeable to carbon dioxide. As a result, carbon dioxide is concentrated in the bundle sheath cell where the Calvin cycle takes place. This high CO2 concentration makes the Calvin cycle much more efficient than in C3 plants. So what I'm talking about here is that we have in the mesophyll cell, so again, this is taking place in two different parts. So you have the light-dependent part of the reaction here, and you have the light-independent part of the reaction taking place here. So that's where it's talking about the light reaction, and the Calvin cycle is happening in the bundle sheath cells. So first, you're going to have your carbon dioxide that's going to come in, and it's going to bond with your PEP. This is going to create oxaloacetate. Then what happens is our oxaloacetate is then going to be, with the help of NADPH, is going to be converted into malate. This malate is then transferred into the bundle sheath cell. Here in the bundle sheath cell, it's then going to combine with carbon dioxide and go through that whole process of the Calvin cycle in order to create its glucose. Once one of those carbon molecules are cleaved from the malate, it then turns into a pyruvate molecule. This pyruvate molecule then moves back into the mesophyll cell, where then with the help of ATP, it's going to turn it back into a PEP, so this whole cycle can happen again. Why is this really efficient? Well, this is because the carbon dioxide is not able to leave the cell. So in here, the carbon dioxide is really, really concentrated. It's also further away from, or because there's so much more carbon dioxide that's found in here, that means that this won't really bind to any oxygen molecules, so which will allow for it solely to combine with CO2 molecules in the area to allow for this process to happen. So again, we can see it in this image here of what's happening, how we have our PP that's going to bond with our carbon dioxide to create malate. This malate is then transported into the bundle sheath cell, where it goes through that Calvin cycle process to create glucose. Out of that, it's going to create that pyruvate, and then that pyruvate, it's going to move back into the mesophyll cell, back into PP. C4 plants use energy to pump carbon dioxide into the bundle sheath cell, where it becomes concentrated. So this method involves preceding the Calvin cycle by first fixing carbon dioxide into a 4-carbon compound. And like C3 plants, C4 plants will close their stomata to conserve water loss. However, unlike C3 plants, C4 plants will continue to make sugar using photosynthesis instead of entering a photorespiration. They do this by initially fixing carbon dioxide in the mesophyll cell via the enzyme PP carboxylate, which reacts the 3-carbon phosphoenol pyruvate, or PEP, with carbon dioxide to form a 4-carbon oxaloacetate, and the resulting of a 4-carbon compound with the mesophyll, then the cycle will then go and enter into the Calvin cycle, where CO2 is highly concentrated to allow for a successful